Good morning, students. So my name is Dr. Gansingh from Government Degree College, Etrunagaram. As you aware, we have the elective paper, the plant biotechnology and tissue culture. So students, in this the technological world, so one of the branch of biology is nothing but plant biotechnology. So day by day, in this world, the population is gradually increased, but the land area is not increased. For all the population, we have to provide the sufficient food materials. But traditionally, it is not possible. That's why we have to adopt some technological methods. So that's why. So number of the scientists, so they work on the technological stream and they developed one of the biological branch that is nothing but the plant biotechnology. In this stream, so it is combined with two sub streams, that is nothing but one genetic engineering and the second one is plant tissue culture. So genetic engineering, it is mainly scoped on using the technology and genes, and they produce a variety of organisms, whether it is animals and plants. So that is nothing but the technology. It is nothing but a recombinant DNA technology. The second one is plant tissue culture. So plant tissue culture is mainly depends upon the very popular term, that is nothing but totipotency. So the totipotency is a capacity of develop the entire plant from a single cell. It is nothing but totipotency. Fortunately, the totipotency, it is present only in plant cells. So 99.9% it is absent in animal cells. If it is also possible in animals, so they can produce their lost organs or lost cells or lost tissues. So students, the plant tissue culture, it is mainly depends upon the totipotency. So first time, the great scientist, the Haberland, so he has conducted several experiments and this, the totipotency, and he's developed the plant tissue culture techniques. So in these techniques, so majorly, once we see the history and the introduction of this plant tissue culture, the culture of plant cells and plant tissues incubated into a synthetic culture medium and under controlled aseptic condition is known as a tissue cultures. So students, so it is not that much of a easy. So we have to maintain the perfect the experiment and we gain the exact result. We have to maintain the aseptic conditions. It is possible only the artificial labs. So we have to make the separate labs for that tissue culture techniques, and it is mainly depends upon the, already I told to you, that is mainly depends upon the totipotency of the cells. The Morgan, the scientist who is first identify the ability of the plant cells is grow as a complete plant under the suitable environmental conditions in 1901. So students, here a single cell, it has a capacity to produce the entire the plant. So it is the first time identified by the scientist Morgan in 1901. Then already you aware and you heard about the two scientists, great scientists, those who are won the Nobel Prize. That is nothing but a Sweden and a Swan. So they proposed one theory. The theory is nothing but a cell theory. So students, the cell theory, it is described only the two mainstreams, the main words, the main concepts. One is the structural and the functional unit of organism is a cell. So it is the main basic of the entire plant tissue culture. So students, here, for example, we take any one plant or any organism or any the animal being. So it is completely meant from, it is made up of entire the body is constructed with the cells only. There is no iron, there is no metal, there is nothing. So all the living organisms, they are constructed or meant by the cells only. And all the metabolic activities, all chemical reaction which takes place in cells only. So that's why here the structural and functional unit of an organism, it is called cell theory, proposed by Sladen and Swan. Later, 1902, the German scientist, the Heberlein, who is now considered as a father of plant tissue culture, he is under the suitable conditions, he is produced the whole plant from the single cell. So finally, the Heberlein, it is considered as a father of tissue culture. So students, here some of the images. So first one is, so already in future, the next class, so we will discuss about that one of the culture, that is nothing but the organ culture. So in plant tissue culture, there are different variety of cultures are there. Cell culture, protoplast culture, leaf culture, shoot culture, organ culture, shoot tip culture. Like this, all different variety of the cultures are included in the plant tissue culture. So the second picture, you can observe that one. It is collect the plant material, simple the process of the mechanism of the root map of the entire the plant tissue culture. So it is very simple. So already we have to prepare one suitable the lab under the hygienic conditions, and we collect some plant materials, which plant we have to produce, we have to experiment. So that plant, first time we are collected from the outside of the laboratory. So that is nothing but that is called in vivo. So we are conducting the experiment under the stable conditions. So that is nothing but that is called in vitro. So after completion of the process, maintaining the scientifically, so the cell, the plant, which plant part we are selected for the experiment. So that is after 2 or 3 weeks, that is ready to grow. Then it is divided and redivided. It is formed a mass of the cells. The mass of the cells is nothing but we are called that is a callus. So after that verse, we are preparing the suitable medium. So primarily we are called the basal medium without any enzymes and hormones. And secondary, we have to add some auxins, some gibberellins, cytokinins like this, all the hormones which is suitable for which organ development. So hormone added the medium we are called that is a nutrient medium. And the third one is the after completion of the all experiment, the cells they are produce the number of plantlets, and the last one also we can see different variety of plantlets. And next image also, so we have observed variety of plants they can develop in the culture vessels. And students, just you observe that image. So there are thousands, lakhs, millions of plants. They are developed a very less area. It is possible with the help of the only tissue culture. It is nothing but it is called, we are called it is micro propagation. So micro propagation, they can provide the maximum number of the leaf plantlets in a very less amount of place. So it is also possible with help of the plant tissue culture. So students, so what is the important objectives of the plant tissue cultures? Let's see one by one. Through tissue culture, large quantities of biochemical substances are produced. So using of this, the plant tissue culture method, we can collect, we can gain large amount of substances which is present in the particular part of the plant. We have to collect and we have to utilize it. The second one, clones are duplicated in short period. Students, for example, so we required large amount of plants in the same way. For example, we take the banana plants. So we have to put 1,000 of plant in 2 hectares or 3 hectares. So for this one, we have to prepare, we have to clone the same variety of plants with help of the plant tissue culture. In a single tray, we can produce the thousands of plants. Next one, bacteria, viral and fungal resistant plants are produced. So students, already the entire process, it is under the control of the lab conditions. So without any bacterial, viral or fungal contamination is not possible in the laboratories. So we produce how they attack and what is their symptoms, what is their characters, we can observe previously and observe immediately, and we can control the such type of the bacterial, viral or fungal diseases, and we can produce the resistant plants. Next one, this technique is used to produce homozygous plants through haploid production. So students all be aware about haploid plants and a homozygous plant. Homozygous means the same type of the plants, and a haploid means one set of chromosomes plant. Already we know very well diploid plants, triploid plants. So haploid, the entire the plant, the each cell, it is contain one set of the chromosomes only. That is called a haploid plants. So using of these haploid plants, we can gain maximum amount of yields and best variety of plants. Next, conservation of germplasm. So we can store, we can maintain the germplasm and we can preserve it. Next, genes are transferred through soma-clonal variants, somatic hybrids, and protoplast fusions. So students, genes are the reasons of the variety of organisms, and we can produce different varieties with the possibility of genes only. Through the soma-clonal variants and somatic hybrids. Somatic means here two protoplasts or two different variety of plants, we can collect their nucleus and we are fused with them and we are creating the somatic hybrids with help of the protoplast fusion. Students, protoplast means here without a cell wall. We remove the cell wall, the remaining part of the cell, it is called protoplast. Using of the fusion of the protoplast, we can produce a variety of plants. Next, to develop gene maps and molecular markers. So students, this is the complete related to the genetic engineering. So which gene is useful for the which character? Already we know very well, students. Here, the particular gene, it is responsible for a particular character. Next, through embryo culture, method seedless plants can be developed. So already the modern days, we are observed number of variety of foods without any single seed. In the olden days, the last 15-20 years back, so we can observe the number of seeds in fruits like papaya, watermelon, grapes, apples, but in modern days without any seeds, so we can consume the entire fruit. It is possible only the help of the plant tissue culture. So students, so basically the plants are developed into the soil in the open atmosphere in the favorable conditions. Favorable condition means here the fertile soil and the well atmosphere, sufficient water and the temperature and sufficient light. It is all the factor, it is decided to the germinate one single plant and it has become a big tree. But here all this thing we have to artificially provided to a plant. So it is possible with help of the different variety of chemicals, hormones, enzymes, whatever. So we have to provide artificially to the growth plant. For this, for growth of these plants in the artificial conditions, so we have to prepare culture medium. So medium, it is basically three different phases: liquid medium, solid medium, semi-solid medium. So medium is nothing but it is composition of nutrients, macronutrients, micronutrients, vitamins, and we have to maintain proper pH also, and add sufficient amount of sucrose for carbon source, and for become it is a semi-solid, we have to add some powder, that is nothing but we have to call agar powder. When we add the agar powder, the entire medium, it is become a semi-solid, like a gel-like structure, and it is easily we have to transplant, which is the explant, means the grown the plant part, that is nothing but explant. So we have to introduce on the surface of the medium. So that is nothing but the transfer of the explant surface of the medium. It is called inoculation. For some best result, we have to add some hormones and vitamins also. So after completion of all this process, students, we have to maintain the sterile conditions, and we have to make the all parts which is used in the tissue culture laboratory. We have to maintain the proper hygienic conditions. Sometimes surface sterilization, sometimes the sterilization process. It is possible with here, students, sterilization is nothing but removal of the microorganisms, means bacteria, virus, in the fungal, whatever, which is attached to the explant. We have to remove all these things, and we have to maintain a proper hygienic condition in the laboratory. So all these, the methods we have to call it is sterilization. So how we sterilized? For example, one by one, dry heat. Students, here we have to use some steel forceps and petri dishes, some glasswares. We have to use all these things. Dry heat, for example, in the hot air oven, already you know very well, oven, the same like we have to maintain one hot air oven in the laboratory. So we have to fix the temperature 160 to 180 degrees. All the material which is kept into the hot air oven, they all are become sterilized. The second one, flame method. Students, 95% of alcohol, using up 95% of alcohol, so we can sterilize with help of the flame, under the flame, or directly we have to apply with rub, with clean with help of the 95% of the alcohol, the surface of the area or in steel parts with help of the 95% of alcohol. Next, students, we are commonly and regularly maintain the one instrument that is shown on the slide, that is nothing but autoclaving. Autoclaving is it is work under the pressure, like a pressure cooker. So students, we have to keep all instruments except the plant parts, which is very delicate, very sensitive, or the cells or tissues or callus, whatever, very sensitive, they just remove outside from the remove and they separated, and the remaining glasswares, medium, and forceps, steel materials, and foil paper, all these things we are kept into the hot autoclave, and once we plug it, we have to wait up to 45 to 1 hour. It is reach 121 degrees centigrade. Then we have to wait up to 15 minutes. Then all are become sterilized, which is kept into the autoclave machine, autoclave compartment. All things they are become the sterilized. So it is very, very important, students, and it is also give visuals also like a pressure cooker which is using in our kitchen. Then students, filtration method. Filtration method, it is also very simply using the filter papers. We have to separate some amount of microbial and any nematodes or any harmful organism using of the filtration process. And already I told to you 70% alcohol method and surface sterilization. Surface mean the upper layer. We are not entering into the deep one we are not open in the part of the which is we taken for the experiment. And when we conduct, where we conduct the experiment, we have to clean each and every part. So that is called surface sterilization. So students, these are some material: scissors, needles, forceps, blades, surgical blades. So these all are utilized for the plant tissue culture, for inoculation, for cutting, for separation, whatever. So we have to use all these things in the tissue culture techniques. So students, just to observe it. So right one is laminar airflow chamber, which is mainly used for the inoculation. So it is give the fresh air and the chamber, the entire chamber, we have to make it very hygienic and very clean. So it is work with help of the ultraviolet rays. There is one light is there. The light, it is killed the ultraviolet rays. They killed all the harmful bacteria or viruses or any fungal members. So that is nothing but laminar airflow chamber. Next, students, here the map shown the collection of the explant and the surface sterilization, washing with help of the water, and then autoclaving, then inoculation in the laminar airflow chamber, and finally the growth is happen in the bottle in the under the aseptic conditions. So students, here already I told to you explant. So these are explant means it is basically two types. One is in vitro and second one is in vivo. In vitro means the taking the plant material which is we are going to use from the existing lab, means it is completely sterile, it is completely fresh, without any, we don't require, not required to, it is again sterile washing and nothing. So it is completely fresh, which is already taken from the laboratory. So that is called in vitro. Then in vivo. In vivo means the outer, other than laboratory, which is available in the fields. So that is called in vivo materials. So students, so finally here the entire process is, it is very simple. Once we see the summary, first we select the plant from in vivo conditions, the take of the plant pot, that is called a explant. Then we sterile all the materials including plant material, then prepare one suitable medium. After preparation of the medium, so we are enter into the laminar airflow chamber and we inoculate the explant under the hygienic or aseptic condition on the upper surface of the medium. Then it is keep for the incubation for 2 to 3 weeks. Later, so it will be divide and redivide with help of the totipotency capability, and it's give the entire plantlets. So thank you students. So if you have any doubts, please contact to me or write any queries to my mail. Thank you once again.